So up next, I'm honored to introduce hold on, I'm joking. Okay, Dr. Valerie Carabetta, an esteemed associate professor of biomedical sciences at Cooper Medical School of Rowan University. Dr. Carabetta earned her PhD in molecular biology from Princeton University and completed her postdoctoral research at the Public Health Research Institute at the Rutgers, New Jersey Medical School. At Cooper, she educates medical students in microbiology and infectious diseases, co-directs the infectious diseases block, and facilitates active learning groups. Valerie's research is at the forefront of combating antimicrobial resistance of bacteria. Her laboratory focuses on understanding protein acetylation of histone-like proteins in bacteria, studying multi-drug resistant Acinetobacter baumannii, and developing novel hydrogel coatings to prevent biofilm-based infections on medical devices. Her work integrates molecular genetics, microbiology, biochemistry, proteomics, and mass spectrometry to address critical challenges in infectious disease treatment. Val is also a past president of our society, so please join me in welcoming Dr. Valerie Carabetta.
Okay, thank you, Jen. Does this thing work? All right. So good afternoon, everyone. I hope you guys are enjoying your day. I know I sure am. And so today I'm going to talk to you about one of my areas of research. We actually have 3 branches in the lab, as Jen was saying, but I'm going to talk to you about my more basic science project, bacterial physiology talk. Just like to say that for those of you who don't know me, again, I'm a basic scientist at heart, which is kind of funny that I ended up at a medical school where my primary job is to teach our future physicians. And I'm not exactly sure what makes me qualified to do that, but if any of you students out there want to know what it's like to be a basic scientist in that world, please talk to me. I'd be happy to tell you about my journey so far. Okay, anyway, so I want to start today by getting everyone starting to think about DNA compaction. Now, it's easy when we think about ourselves and human cells. We have all these long linear chromosomes, meters of DNA that somehow are going to get shrunk down and fit inside the nucleus of our cell. But what about bacteria? So as we all know, bacteria have 1, maybe 2 circular chromosomes. And so at first glance, it seems like that's not really a problem. However, the bacterial chromosome still needs to be compacted at least 1,000-fold to fit inside the tiny nucleoid region of the cell. Okay, so here I'm showing you a classic famous image of a lysed E. coli cell with the DNA negatively stained. And what do you see when you look at this structure? You might be expecting to see an unorganized, tangled ball of yarn, but in reality, this is referred to in the field as a rosette model, where let's see if this pointer is going to work. Where are you? Okay, a little pointer here. All right, we'll work with this arrow. So what you can see in the middle here is that there's a densely staining core, and out of that core radiates hundreds of topologically isolated supercoiled loops. Now, in bacteria, this superhelicity and supercoiling is the first level of DNA compaction. And the rest of the chromosome is compacted and further organized through the action of a special set of proteins referred to as nucleoid-associated proteins, or NAPs. So here is a lovely artist rendition of a eukaryotic histone. We're going to jump back to eukaryotes for a second, and then we're going to dive hardcore into bacteria. So as most of us probably know, the histone is made up of an octamer, 4 histones in duplicate. So we have an octamer called the nucleosome. The DNA is wrapped around that approximately 2 times. And what I really want to bring your focus to, though, is these dashed lines here. These dashed lines are representing the unstructured N-terminal extensions of each of the histones, referred to as the tails. And these tails are rich in basic amino acids and are the site of various post-translational modifications. The combination of these modifications is thought to be regulatory in nature and are often referred to as the histone code. So I'm going to show you an extremely oversimplified version of how these post-translational modifications, or PTMs, are believed to regulate histones and DNA, like gene expression and other transactions. Okay, so in our unmodified state here, the histones are tightly associated with the DNA, leaving no room for RNA polymerase or other transcription factors to find their promoters and activate them. However, if these are modified by a PTM, say acetylation, this weakens the association of the histones with the DNA, revealing various promoters and genes and allowing for transcription to occur. In addition, there are other proteins that can bind to acetylysine residues and further modify the chromatin, as well as recruit or serve as additional transcription factors. So I'm going to talk to you today about the most widely conserved map in bacteria, referred to as the HU family, and there are orthologs in every single bacterial species. So this is a crystal structure that was solved from Phoebe Rice's lab, and I really like this one because it's one of the only ones that actually has the DNA density in the structure. So if you look over here, so that they bind as dimers, and what I really want you to take away from this image is look at that incredible bend. So it's a greater than 180-degree bend that's introduced upon binding of these molecules to the DNA. Now, so it's this bending action that's believed to organize and compact the bacterial chromosome. In Bacillus subtilis, which is our model organism of choice, the ortholog is referred to as HBSU. And HBSU is essential. That's not true in all bacteria. And it also compacts the DNA and is known to be involved in various DNA transactions, including DNA replication, initiation, recombination, and repair. And if that sounds remotely familiar to you, well, that's sort of the functions of the eukaryotic histones. They compact DNA and regulate DNA transactions. So the HU family of proteins is generally thought to be functional homologs of eukaryotic histones, although they have no sequence conservation or structural conservation. But what is known is that they all bind as dimers. In gram-positive bacteria, including Bacillus, they bind as a homodimer, where that's not true. In a lot of gram-negatives, they actually bind as heterodimers, and they bind non-specifically to DNA, but have a preference for AT-rich regions, as well as structured DNAs, including bends, kinks, or breaks. So I got interested in this protein number of years ago. God, it's like 12 years ago when I probably started thinking about this protein, when I was characterizing the Bacillus subtilis acetylol. And what I found from that work is that HBSU is acetylated at seven sites, and these are represented here as the diamonds in this schematic. And perusing through the literature, there's a lot of large-scale proteomic studies out there looking at various post-translational modifications. Also, HBSU came up as phosphorylated at a threonine at position 4, as well as arginine phosphorylation at position 61. And another paper characterized the succinylol of these of Bacillus subtilis. It was found that some of my lysine acetylation sites are conserved between these two, and site 1875 and 86 may be either acetylated or succinylated. So taking a step back, can't help but wonder if what we're looking at right here is evidence for the existence of a histone-like code in bacteria. So we expect two things are going to be true if there really is some kind of histone-like code. One, well, there really ought to be some kind of functional significance to these acetylation sites. And two, there needs to be a mechanism to put these groups on and take them off if it's truly going to be regulatory in nature. So for the remainder of my talk, I'm going to first tell you about our work, what we've done to explore the functional significance of acetylation, specifically with regard to the process of sporulation, as well as antibiotic survival and persistence. And then I'll give you a brief update on some of the things that we're doing to study the regulation of HBSU acetylation and the specific mechanism. Okay, so before I dive too deep into this, let me tell you how we study acetylation. Using substitution mutants in vivo. So up here we have lysine and acetylysine. To mimic the deacetylated state, we mutate that lysine residue to arginine. Now, this maintains the positive charge of the lysine residue, but it itself cannot be acetylated. To mimic the acetylated state, we mutate the lysine residue to glutamine. And glutamine is a neutrally charged amino acid. And I am aware that these do not look exactly the same. But these are generally accepted in the field. And there are countless examples in both eukaryotes and prokaryotes, in both in vivo and in vitro, that the glutamine substitution recapitulates the phenotype seen with acetylysine. Okay, so when I was still a postdoc in David Dubnow's lab, one of my tasks, I started studying these, and I actually made all 14. So we have 7 sites. I made 14 lysine to glutamine, lysine to arginine substitutions in the chromosome at the native locus. And one of the first things I did was just examine these. And so up here is what the bacillus cells look like. They're just rods. They're not very exciting. And you can't see this great because of the lighting in here, but that looks like green rods. It just kind of looks like the nucleoid takes up the entire cell, and that's DAPI stained. So when you look at the deacetylated mimic, so the R mutation, you can see that the nucleoids are smaller and much more rounded and are more compacted. And if you look at the opposite mutation, this is not the case. They're there, trust me, the lighting is poor in here, but they look much, maybe somewhere in between, but they still have that oval shape and don't look very, very compacted. And this phenotype I observed in 6 of 7 spots. And a way to start thinking about this actually comes from an examination of the structure. So this over here is a structural model of the of HBSU. And so that, remember, it's a dimer. So one of the dimers is this cyan color, and the other one is purple here. And all the protein acetylation sites are labeled here. So if you look at the rotating thing next to it, you can see that's where the DNA molecule would be. And so if you picture where the DNA molecule will be on this one, where all the sites are labeled, the DNA is going to sit in these arms. It's going to extend out of the screen and wrap down in front of the molecule, and extend back into the screen and wrap down behind it. And if you can imagine where that is, you can see that many of these residues are in position where they might make direct contact with the DNA. And so I told you 6 of 7 show that nucleoid compaction phenotype. And the only one that did not is K37R. And if you look at the structure here, you can see that the K37Rs are most definitely pointing out and away from where that DNA molecule will be. So this suggests that most of the sites are making direct contact with DNA, and they're needed to compact the DNA. And that acetylation of these sites modulates and somehow controls what's going on in a wild type cell. All right. So now I want to switch gears and tell you a little bit about sporulation. You can watch this fun little sporulation movie up the top to give you a couple of important highlights down here. So as we all know, bacteria don't live a very exciting life. They grow, they get longer, then they split in half and there's 2 identical cells. However, some bacteria, such as Bacillus subtilis, can enter a survival mode, referred to as sporulation. It has a special developmental program within its genome to do this. And what happens is that when the cell is starved and really, really hurting, this is a last-ditch survival effort, they will enter the sporulation program. And the hallmark feature to start this program is that they divide asymmetrically. So the septum does not form at mid-cell. And the smaller compartment here is eventually going to become the mature spore. And as the process continues, there's a very complex crosstalk that's going on with genes and regulatory factors. I'm not going to get into all those details. But in the end, we have the mother cell sacrifices herself. So that's why it's a last-ditch survival effort. And the spore is super protected from all sorts of environmental insults, including desiccation, UV stress, chemical stress, heat stress, and it can persist out in the environment for a number of years, including maybe even decades. What's cool is they've actually brought Bacillus subtilis spores into space, and they brought them back, and they're just happy and fine. Okay, so we started to look to see if acetylation of HBSU impacts the sporulation process in any way. So most of us will always start by looking at sporulation frequency. So the cells are grown for 24 hours in a special media that promotes sporulation. It's called DSM. And we then, the next day, you heat the culture to 80 degrees, only the spores will survive, and any vegetative cells will die, and then you can calculate frequencies. So wild type cells are sporulated in this background in our media, about 65–70%. And you can see that when we make all the acetylmimic mutations over here, that there are large defects, with the exception of this K41 strain, and we're going to come back to that in a little bit. And then on this side, the R mutants are over here. A lot of them have defects too. Again, K41 is a little bit different. We'll talk about that in a minute. Well, in cases like this, these are opposite mutations where we have R looks like wild type over here, whereas the K3Q looks very much so, has a huge defect. So we would speculate that, well, for sporulation, K3 should be mostly deacetylated. It's not maybe as obvious what happens when they both have a phenotype, which is something frequently seen, but we really don't know what the actual stoichiometry of these things in the cell is, as our mutants are locked in 100% acetylated or 100% deacetylated. But what I'm going to tell you about in the next few slides kind of sheds a little bit more light onto what's going on in these different backgrounds. Okay, so in this paper, this is one of the first papers from my lab from 2022. The sporulation is not zero, so we were able actually to isolate those spores and look at their spore properties. So not only did most of these strains not make spores at the proper level, the spores that are made aren't good at being spores anymore, and they're more susceptible to heat, chemical stress, or UV exposure. So this first work established that HBsU acetylation is playing some kind of role, but we didn't actually figure out the mechanism of how that's working. And this is some new work was carried out by two former students in the lab, and Olivia and Reticia, and my PhD student, Leah. We are actually getting ready to publish this story right now. So if anyone's interested, be on the lookout for it. And for this work, we looked at the possible contribution that HBsU acetylation is regulating gene expression. But before I talk to you about the data, let me just explain a few things about this. So for this, we grow spores in that sporulation promoting media in DSM, and we take samples at what I'm going to call T0, T1, T2, and T4. T0 represents the transition between exponential and stationary phase, and it's when the decision-making process is going on, where the cells are analyzing their environment, and if things look bad, they're going to enter into the sporulation program. So T1 would be 1 hour after that, T2 is 2 hours, and so on. And each one of those is corresponding to different morphological stages. So our early sporulation events are occurring at T0 and T1, and then our later ones are T2 and then T4. So we took samples at these 4 time points, isolated RNA, and then performed quantitative real-time PCR. So we actually analyzed 25 key genes of important transcription factors and regulatory factors throughout the entire sporulation program. And in one mutant, this HBS K41Q mutant, we found that 17 of 25 of those genes were significantly upregulated, especially at T2 and T4 time points. And I'm showing you some of that data here. And this is the quantitative real-time PCR data. And what we're looking at here is the mutant normalized to the wild type. So we're looking at how this changed compared to the wild type. So everyone can envision wild type is set at 1, and you can see how it varies throughout the time courses. So SIG-F is a sigma factor. It's a 4-spore specific sigma factor that's key to keep the developmental program going. And you can see at T2, it is highly overexpressed. And we also found that the protein levels are also overexpressed. And then the consequence of having more protein is that there's more gene expression of its target genes, and that happens to be what this SPO2Q gene is a direct reporter for SIG-F activity, as it is completely dependent on only SIG-F activity. So what this looks like then, I think I have some more data. Yeah, so here's another data. We actually did analyze the opposite mutation because we had so many changes happening here. And this is a bunch of showing you at the T2 time point, a bunch of our genes that we analyzed, and you can see a lot of them are highly and significantly overexpressed in this background, whereas in the opposite mutant, they either are wild type or are slightly less than what you would see in wild type. So all these changes got us wondering is, do these guys enter sporulation earlier than they're supposed to? Or maybe they also are going through the program faster than they're supposed to without the appropriate environmental cues. And so one way we looked at this was we looked at T0, which is a time when you shouldn't find many spores because most of the cells are just deciding if they want to enter this program. And so what we found was that in wild type cells, you can find very few, where it would be like 0.005% kind of thing. Or maybe a little higher than that, maybe 0.05. And then where's my mouse? And then if you look here in the K41Q background, you actually can detect them at average for our replicates of about like 0.6, which is a 25-fold increase. I should also mention something else we started thinking about as we thought about what's going on in this K41Q strain. The K41Q spores were more susceptible to heat than wild type, which means that as heat is important in our assay to determine these frequencies, we are actually underestimating the amount of true sporulation in these strains. And so what we think from these findings is that K41 acetylation, first of all, it seems pretty clear that activates gene expression of a number of genes. And we think that this represents an on-off switch that helps the cells transition from the early to late stationary phases. So, because I want to tell you guys a couple of other stories, I didn't have time to show you, we actually also looked at the other sites and they had the opposite phenotype. They actually led to decreased transcription of many of the key regulatory proteins. And then finally, something interesting that really is like as driving our exploration of these enzymes is that if this is some kind of on-off switch, if it's like acetylated early, then gets deacetylated to switch the program around, well, then that means there needs to be sporulation-specific enzymes. There needs to be some acetyltransferases and deacetylases that are activated at specific times during the sporulation program, which is something that we are extremely interested in following up on. Okay, so I want to tell you a quick another story before I tell you about the enzymes. So this is done by two former students in the lab, for Trish and Rachel, and two of my current students actually participated in this, and Precious and Brianna. And the idea for this study came out of from Mycobacterium. So there actually is a high interest in the HU family protein in Mycobacterium, including acetylation. And there was a paper that said that reported that Mycobacterium smegmatis, when exposed to an antibiotic called fusidic acid, the response of that bacteria was DNA compaction. Now, as Mycobacterium and gram-positive bacteria, they're not exactly the same. We wondered, is this kind of a universal response to antibiotic exposure? What happens in Bacillus subtilis? So not sure how well you can see these microscopy images. I'll just tell you what you see what you can see in our quantification below. When in the presence of the drug kanamycin, you see those small dots again, those small, instead of the oval shapes, so that they also compact their nucleoid in response to antibiotic stress. And here in this like salmon color here, this is measurements of nucleoid area that we've used our microscopy program, our program to do. So the salmonish color is wild type. And then in the presence of kanamycin, you can see here, this is the blue. So this is a cumulative distribution plot. It's actually related to the histogram, but it's a little easier to see patterns when you plot your data like this. And you can compare the medians. It's just the 50th percentile here. You can see shifts. And so as you can see, that blue line is shifted to the left, which means the population is distributed more towards a smaller nucleoid size. So we analyzed all of our mutants and 3 of them stood out. So the K3Q and K75Q strains, they didn't do anything. They didn't compact their nucleoid in response to antibiotic stress. As you can see, the 2 lines plus and minus drug overlay each other. And the other interesting mutant was again K41. K41Q strains, instead of making their nucleoids smaller, it actually seemed to get larger. And where's my mouse? So in these pictures, blue is no drug, red is with drug, and you can see the curve is shifted the other way. Okay, so we thought that, well, maybe the ones that can't compact their nucleoid, maybe that they're gonna die faster. In the presence of drugs, we did time kill assays, and it turns out, well, they do, but all of them do. Which suggests that, well, nucleoid compaction isn't saving them from killing from the antibiotics, and perhaps there's another role, something else is going on in these strains. And given our results with sporulation, they're also probably affecting gene expression. That's something that we need to follow up on. Okay, and just quickly, a little note about persistence. Again, in Mycobacterium smegmatis, a HUP-B, which is the HU family ortholog in that background, that K86R, so we have a conserved site between the two, and that's the deacetylated or unmodified mimic mutant. And if you do that, it lost a specific subpopulation of drug-tolerant bacteria upon isoniazid exposure. So this got us thinking a little bit about bacterial persistence. And for those of you who are not as familiar with what bacterial persistence is, so normally most of the bacteria in your population will be susceptible to a drug. And when you add drug, they die. If you take it away, they're gone, they can't come back. Persisters are evolutionary cheaters, where they grow very, very slowly or practically dormant, so that when challenged with drug, they actually survive because they're just sitting there not growing. And then when you take the drug away, the bacteria return. Eventually, these persistent subpopulation will appear again. And you can see that over here on these pictures over here, where you see the susceptible ones die, nothing ever comes back. But if there are persisters present, they will die, and then they will persist, and you take away the antibiotic and they can grow again. So we grew our cells in the presence of antibiotic for over 6 hours. Most of the cells are going to die, then we wash away the drug and let them come back. And I want to show you the data with the R mutants. So on the left, you have kanamycin exposure. On the right, you have vancomycin exposure. And you can see the black line. Where is that pointer? Here it is. The black line is the wild type cells, which they start growing pretty quickly. Recover, and the 37R strains are pretty good. All the other ones are pretty slow to recover, especially K18R and K41R. And the same thing over here, you can see the 37R pops up a little bit before wild type in this case, but all the other ones are shifted to the left, and K41R is really, really delayed in recovering from the persistent state. And if you remember, I told you before, K37R is the only ones that don't compact their nucleoids. So when the nucleoids are compacted, that's likely compatible with the persistent state, but they can't escape the persistent state. They must need to decompact their chromosome in order to escape. So this allowed us to propose the following model, where if there's some acetylation pattern during normal growth, things are fine. Antibiotic stress, then we would need to propose that there is a lysine deacetylase that gets induced, and it will deacetylate, especially at K3 and K75. Those are the two mutants that did not respond properly to antibiotic stress. And then we would propose that a lysine acetyltransferase would then be induced, acetylate at least starting at K41, and then that would allow the cells to recover from the persistent state. Okay, so just briefly, another couple of minutes, just want to tell you what we're doing so far with the regulation of acetylation. So just quickly, in Bacillus celis, here's what our picture looks like so far. So we have proteins that need to be acetylated. There was, prior to our work, one known acetyltransferase called ACUA. So it takes the acetyl group from acetyl-CoA and it acetylates the lysine residue. This protein seems to be exclusively involved in regulating enzymes that form AMP intermediates, such as acetyl-CoA synthetase. There's two known deacetylases in this species. We have ACUC, which is a hydrolase that uses zinc as a cofactor and acetate as a reaction product. And then there's the SIRT2 and SIRT-N, which uses NAD as a cofactor, and the reaction products are nicotinamide as well as 2-O-acetyl-ADP ribose. So early on, for my later years of my postdoc, early years of starting my own lab, we showed that one acetyltransferase, YFMK, directly acetylates HBSU, both in vivo and in vitro. This is an in vitro assay using C14 labeled acetyl-CoA, and you can see as we increase the amounts of YFMK, we get an increased signal here, and if we have two mutations that kill the activity of this acetyltransferase, we no longer get acetylated HBSU, and we actually confirmed that with mass spectrometry. We could look at the site specificity of this enzyme and saw increases in the presence of enzyme, especially at these C-terminal residues. And since this time, we now have evidence that there are 3 more HBSU acetyltransferases, including YDGE, YDHI, and YACD. So this work was started by my former postdoc, Ola, and is being continued by my current postdoc, Mohammed. And you can see here what our preliminary data is suggesting. If this pans out to be true, that YDGE seems to really like K41, that would be my suspected one that's going to be induced during sporulation. But again, this data is preliminary. Mohammed is currently investigating a very interesting thing we found, in that these 3 new acetyltransferases autoacetylate. And we're trying to understand, what does that do? Does that increase or decrease the activity? Because it's a way to regulate these guys, whereas YFMK does not autoacetylate, which is something that's interesting for us to follow up on. Finally, we're also very interested in which one of those 2 or another one deacetylates HBSU. So hopefully, some of you got a chance to talk to Maxwell at his poster today. He was telling you about the inhibitors. I don't want to overlap with his stuff too much. But one thing we are going to do is develop a new assay to study HBSU deacetylation in vitro. So we're going to use a YFMK that's tagged with GST. We can remove YFMK then with magnetic beads and get acetylated HBSU, where we will mix it with a sirtuin plus NAD and analyze by mass spectrometry to see if this is in fact the enzyme that deacetylates HBSU. We also have some good in vitro assays going. This is some data that was generated using actually a histone H4 acetylated peptide, and you can see SIRT-N has strong activity for that. You see here the M over Z values, a loss of a minus 42, which is the size of an acetyl group. And we have an ACS A peptide that's a known substrate, so that's our control, and we actually are also generating some HBSU acetylpeptides to run with this. And then if any of you didn't get a chance to see Maxwell, he's analyzing and working with inhibitors of this enzyme activity, so we could probe the significance in vitro and possibly develop a novel antimicrobial agent for the future. Okay, so told you today, I found there's 7 acetylation sites, at least 7, and many of them contact DNA. 6 or 7 of them are needed for proper chromosome compaction, except that one that was pointing out. Maybe that's in protein-protein interaction phase. For sporulation, this K41 site, especially in these stationary phase things, seems to be very important. We think that it's an on-off switch that allows the cells to transition from early to late stages. And we also, I didn't show you the data, but that think that deacetylation of some of the other sites may be required to turn on gene expression of specific genes. K3 and 75 seem to be like they need to be deacetylated for proper nuclear compaction during antibiotic stress. And that K41 might be the start of a signal that they can re-enter growth, and possibly by decompacting the chromosome following antibiotic challenge. We have at least 4 acetyltransferases now. We still need to figure out when they're active and when they deacetylate this protein. And we also need to determine which one is the HBSU specific KDAC. If I had to guess, it's the sirtuin, because the sirtuins in other species seem to regulate the HU family protein. Okay, with that, I want to thank my lab here, as well as my collaborators at Princeton, the Ileana Christia's lab. And working with Maxwell is Brian Weiser. He's an expert in sirtuin biology, and of course the NIH for funding. And thank you all for your time. I'd be happy to take any questions. Yes.
Very nice. Are the genes in code that we see transfers?
Individually, we can knock them out. We have not studied doing all of them yet, which is something that we need to do. We've only done 2, which seems to be okay, at least in LV, but we haven't really checked in other medias that might be a little bit more relevant.
Well, I guess what I'm wondering is, in acetyl-CoA cell acetylate or acetylphosphate, so is there a metabolic signal that would lead to something like change in sporulation? So could you like knock out the synthase, have acetyl-CoA phenolate, or knock out acetyl kinase, you would find out phosphate levels to see if you see phenotypes through formulation or akin resistance.
Yeah, we could do that. Acetylphosphate is a known substrate for non-enzymatic acetylation in bacteria. But we believe that HPSU is mostly enzymatic, but that's something we can certainly look at to see how it playing with the levels of these different metabolites in our species, if they can also regulate, because I wouldn't be surprised if there's a metabolic signal to change things.
Well,
Hello?
is there any kind of genetic thing going on, like, cytosine methylation on, bacterial DNA, like there is on eukaryotes, in this bacterial system?
I'm not aware of any like cytosine methylation like an equivalent, but that doesn't mean there isn't. I wouldn't be surprised if there was something like that, because I feel like well, I always like to say that bacteria are people too. And a lot of the things that we discover in ourselves turns out to have a different version in bacteria. But I don't believe there's any equivalent known right now of the CPG. Hello? Is that on?
Let's call that.
Okay.
So, histones and eukaryotes are like super well conserved, like across organisms. They're very, very, very well conserved. In bacteria, is that also the case? Like, does the DNA tend to fold around the bacterial histones in the same way, or is it really different for species? And like, do the lysines line up in similar places across the?
Yeah, that's a good question. Do I have that here? It's on. Oh, go. So yeah, I actually did look at this. So yeah, so first of all, the HU family proteins, every bacteria has them. They're not always essential because a lot of the like E. coli, for example, has 2 versions of them. But if you knock out both of them, the cells are very, very sick without them. Positive, they seem to have less of these naps, so that's why I think that they might be essential in these particular bacteria. But yes, these positive residues are very highly conserved. So if you kind of just scan through, I know it's a little small, but a lot of the Ks through here are conserved. And what I'm actually, this is from one of our review paper I wrote, all these red things are actually the acetylation sites. And it seems like the acetylation sites themselves are also somewhat conserved. There's a few like hotspots, like the C-terminal region has a lot of acetylated sites in different vast species. So, and this is looking at all sorts of gram positives, gram negatives, as well as mycobacterium. And so I think that this is a widely conserved mechanism throughout bacteria. And the structural homology, if you think back to that figure I showed you with that was actually 4 different species of them lined up. The structure of this family is so highly conserved that I think that the mechanism of the DNA wrapping around it is going to be identical in all of the bacterial species.
Any other questions?